From the dawn of civilization, humans have marveled at the diversity of life on Earth. We observed the inheritance of traits, the passing down of characteristics from one generation to the next. But it wasn't until the 20th century that we truly began to understand DNA. This molecule holds the blueprint for every living organism. With this knowledge came the power to modify genes. This is the essence of genetic engineering. It's about shaping life and addressing humanity's challenges. Imagine a world free from genetic diseases where crops withstand harsh climates and bacteria clean up pollution. This is the promise of genetic engineering, but it comes with profound responsibilities. Altering life raises ethical questions. We must proceed with caution and respect for nature. This is our journey into genetic engineering, to explore its tools, applications, and ethical dilemmas, understanding the power and responsibility it brings. Imagine you want to build a house. You wouldn't just start piling bricks randomly. You'd need a blueprint, a set of instructions to guide your every step. In the world of biology, DNA is that blueprint. It contains all the information needed to build and maintain an organism. The field of genetic engineering wouldn't be possible without the ability to manipulate this blueprint to cut, paste, and copy specific genes. This is where DNA cloning comes in. This fundamental technique allows scientists to create multiple identical copies of a DNA segment, much like photocopying an important page from a book. The process begins by isolating the desired DNA segment, the specific gene of interest. This DNA fragment is then inserted into a vector, a molecular vehicle that carries the gene into a host cell, often a bacterium. As the host cell replicates, it also copies the foreign DNA, creating numerous identical copies of the desired gene. DNA cloning has revolutionized biological research and paved the way for countless applications. It allows us to produce large quantities of specific genes for study, to create genetically modified organisms with desirable traits, and even to develop life-saving therapies like insulin production for diabetics. This ability to replicate genetic material is the cornerstone of modern biotechnology. The discovery of restriction enzymes in the 1960s was a watershed moment in genetic engineering. These molecular scissors, naturally found in bacteria, provided scientists with the ability to cut DNA at specific sequences. Imagine a pair of scissors that can locate a specific word in a book and snip it out with incredible precision. That's the power of restriction enzymes. Each type of restriction enzyme recognizes a unique DNA sequence, its target site. When it encounters this specific sequence, it binds to the DNA and cleaves it, creating either sticky ends which have single-stranded overhangs or blunt ends, which are straight cuts. These cut DNA fragments can then be joined with other fragments that have matching ends, creating recombinant DNA molecules. This ability to cut and paste DNA fragments with such precision is fundamental to genetic engineering. It allows scientists to isolate specific genes, to study their functions, and to create novel combinations of genetic material. This technology is used in gene therapy, where faulty genes are replaced with healthy ones, and in the development of genetically modified crops with enhanced traits like pest resistance or increased nutritional value. Restriction enzymes truly are the precision instruments of genetic engineering. Cutting and pasting DNA fragments wouldn't be possible without another crucial tool, DNA ligases. This molecular glue plays a vital role in joining DNA fragments together, ensuring the stability of the newly formed recombinant DNA molecule. Think of it as the glue that holds together the pieces of a puzzle, creating a complete picture. DNA ligase forms a phosphodiester bond between the sugar and phosphate groups that make up the backbone of DNA. This bond seals the nick in the DNA strand, permanently joining the two fragments. The process is highly specific, ensuring that only compatible DNA ends are joined together. The discovery and application of DNA ligase were crucial steps in the development of genetic engineering. This enzyme allows scientists to create functional recombinant DNA molecules that can be introduced into host organisms. It's used extensively in gene cloning, where DNA fragments are inserted into vectors and then replicated in host cells, and in the creation of transgenic organisms, where genes from one species are introduced into another. Without DNA ligase, the field of genetic engineering as we know it wouldn't exist. Imagine having a life-saving medicine but no way to deliver it to the cells that need it. In genetic engineering, getting the desired genes to their destination requires a special delivery system: vectors. These molecular vehicles are designed to carry foreign DNA into host cells, where the genes can be expressed and their effects observed. Vectors come in various forms, each with its own strengths and limitations. Plasmids, circular DNA molecules found in bacteria, are commonly used in laboratory settings for gene cloning. Viral vectors, on the other hand, are engineered viruses stripped of their harmful properties and repurposed to deliver genes into specific cell types. They are often employed in gene therapy and vaccine development. Regardless of the type, all vectors share essential features. They need an origin of replication, a DNA sequence that allows them to replicate within the host cell. They also require selectable markers, genes that allow scientists to identify and isolate cells that have successfully taken up the vector. And finally, vectors need multiple cloning sites, regions of DNA where foreign DNA fragments can be easily inserted. Vectors are the workhorses of genetic engineering, enabling scientists to deliver genes into cells for a wide range of applications. From producing therapeutic proteins to developing genetically modified crops, vectors play a crucial role in harnessing the power of genetic information. Genetic engineering is not a futuristic fantasy, it's a reality that is already transforming our world in profound ways. From medicine and agriculture to environmental science and biotechnology, the applications of this powerful technology are vast and ever-expanding. In medicine, genetic engineering holds the key to treating some of humanity's most challenging diseases. Gene therapy, for example, uses engineered viruses to deliver functional genes into cells with defective ones, offering hope for patients with genetic disorders. Scientists are also exploring CRISPR-Cas9 technology to develop targeted therapies for cancer and other complex diseases. Agriculture has also been revolutionized by genetic engineering. Genetically modified crops, engineered for traits like pest resistance, herbicide tolerance, and increased nutritional value, are becoming increasingly common. These crops have the potential to enhance food security and reduce reliance on pesticides and herbicides. Beyond these fields, genetic engineering is being used to develop biofuels, create biodegradable plastics, and clean up environmental pollution. It's even being explored as a way to conserve endangered species and restore damaged ecosystems. The field of genetic engineering is constantly evolving, with new discoveries and innovations emerging at a rapid pace. The future holds even greater promise for this transformative technology, with the potential to reshape our world in ways we can only begin to imagine. CRISPR-Cas9, a revolutionary gene editing tool, continues to advance at an astonishing rate. Scientists are developing more precise and efficient CRISPR systems, expanding its applications in medicine, agriculture, and beyond. The technology is even being explored for its potential to correct genetic defects in human embryos, raising profound ethical questions about the future of human evolution. Another exciting area of research is synthetic biology, where scientists are using the building blocks of life to create entirely new organisms and biological systems. Imagine designing bacteria that can produce biofuels, clean up toxic waste, or even manufacture pharmaceuticals. The possibilities of synthetic biology are limited only by our imagination. As genetic engineering continues to advance, it's crucial that we proceed with caution and responsibility. The power to alter the building blocks of life comes with profound ethical implications that we must carefully consider. The ability to manipulate life raises profound ethical questions. Genetic engineering holds promise for health, agriculture, and the environment, but also presents risks and dilemmas. Human germline editing is contentious, as changes pass to future generations, raising concerns about unintended consequences. The debate over designer babies raises concerns about eugenics and social inequality. Addressing these challenges requires public discourse, regulatory frameworks, and dialogue between scientists, ethicists, policymakers, and the public. As we venture into genetic engineering, we must proceed with wisdom, caution, and respect for life and nature.